それでは小林様。
So Dr. Kobayashi, please.
Good afternoon. First of all, how have we been promoting innovation in the medical field? I use antibodies and target cancer in the treatment. And as you can see, there is the methodology of using antibody itself, but cancer isn't fixed. So far, things were attached to antibodies to target cancers, but the situation hasn't improved. And we've tried everything that's on this diagram, and yet we haven't been so successful. And this methodology was created 25 years ago. You may think that medical innovation takes such a long time, but this was developed 25 years ago, and a radioactive isotope is attached to the antibody and it's injected, and then it's concentrated to the cancer cell, but it also exposes other good cells to radiation. So my innovation is to do something to the antibody, but then inject avidin, which is indicated here. Then all the antibody which isn't concentrated on the cancer is trapped by the organ, and this isotope is released. And as indicated on the fourth diagram, this would only target the cancer cells. This is an old diagram I drew 25 years ago. This is the mouse, and here you see the T. And we were able to develop this methodology that would target only the cancer cells. And this was created as a radioimmunotherapy 25 years ago, which targets only the cancer cells. And I brought this to the United States thinking that this will offer much treatment for cancer patients. But this wasn't realized. Why not? Because at the first injection, as indicated, the whole body will be exposed to radiation. And during the first 2 days, it will also expose the other parts of the body. So you can't increase the dose to huge volume, and it's therefore going to be less dose than is necessary to kill the cancer cells. And already radiation therapy is being done for cancer treatment, but 60 gray or 80 gray is the unit of dose that has to be used to kill cancer cells. But all the immuno cells would be killed at a fraction of that. So if we increase the dose to the level that it would kill the cancer cells, it would also kill the immune cells. And secondly, there could be the second drug to clear this problem. But then you would have to expose the patient to 2 drugs. And even 1 drug is difficult to be certified and approved. And then if we need the second drug, then it would be very, very cumbersome to establish a new therapy. And the second drug was a hen egg, so people with egg allergy cannot be able to take this medication. So what could be done? These were all constraints. So unless we solved these limitations, we would not be able to establish a cancer therapy. So this was the idea. Non-toxic agents will be used. Nothing that would be toxic to the body will be used, because there would be constraint to the dose if we use something that is toxic. And the second factor is not to use radioactivity, because it is toxic. And if radioactive substance is used, then it would be toxic. So near infrared, let's use something that is visible. So impose the photoimmunotherapy to only the cancer cell. And we came up with this technology, and to give the long story short, this is the kind of treatment that we have developed, and this is what we are trying to do. An antibody, the chemical substance is attached to the antibody and is injected into the patient's body. And in a day or two, the antibody would flow and reach the target cancer cell. But it doesn't attach itself to healthy cells. And only the cells with the antibody will be killed with the light. And there will be the swelling of the cell and will break up. And only the cancer cell will explode and the healthy cells will remain.
Causes the tumor to shrink, creating spaces between the cells. The tumor will die and normal tissue will regrow in its place. Photoimmunotherapy is a new form of cancer therapy that is highly specific for cancer cells and does not damage nearby normal cells. It is currently in clinical trials in patients with inoperable tumors.
So, this is the therapy we developed, and 2 years ago, we obtained FDA approval. So, therapy is now being administered. Clinicaltrials.gov is the site where all of this information is accessible. So, currently, this treatment is given to head and neck cancer patients.
Dr. Stenson issued this news release, and if you pick up from there, if you apply light, right afterwards, the cell starts dying and it turns white and melts away. The surrounding cells are not damaged at all. It's very safe. That is what she says. Whether it's animals or human beings, I think it's the same. If you apply light, in the middle you see that it turns white and it dies and melts away. It's a very quick process. Mary Will, who is the top responsible doctor, he says that this therapy is very safe and it is being applied very safely. This is just a cartoon. In this way, the cell swells. In 1 minute, in this way, the cell bloats and it bursts. In a minute, it swells and it bursts. It's a very quick physical reaction. And the good part about this is that the surrounding cells are all alive. So in this way, the cancer cell breaks, but immunity works. Immunity cells don't die. They can provide the immune functions. And the other, it can induce immunity as well. If you treat just the portion in red, those samples that are not treated are effectively treated. If you plant 4, if you treat 1, all of the tumors disappear. In the case of the mouse, that was clearly done. And this is a very cell-specific. If it's the same sample, it all dies. And it has no impact on normal cells. That means, in this way, if you break the cancer cell, the immunity cells know that this cell has broken. They know that that is the broken cancer cell. And so it is the immunity cells are educated as to how to attack the cancer cell. What had stopped the immunosuppressive cells, if you remove these cancer cells, it will clear the other cancer cells, and metastasized parts are also treated. We use antibodies. The target, there are several targets, and to the target, if you create antibodies, you can treat many patients. But the cancer cells that break have grown in each patient, in each respective patient. So the immunity for these broken cancer cells differ between patients. So it's not one drug being applied to a lot of patients. It's a precision medicine. It's individualized medicine. In a tailor-made way, we treat cancer patients. So that is the best method, I believe, to cure cancer. This is the member, as you can see, Spirian. And Mikitani-san is the largest investor, and so we all work together as a group. Thank you very much.
小林様ありがとうございました。
Dr. Kobayashi, thank you. We're going to be joined by Etsuko Nagayama of the Editing Bureau of Mainichi Newspaper Company for a dialogue. Good afternoon, ladies and gentlemen. My name is Etsuko Nagayama of Mainichi Newspapers. You're probably wondering why I am sat here. 10 years ago, in 2007, I got to know Dr. Kobayashi's research, and since then I've been following his story. When we newspaper journalists write medical articles, we ask them in what year their technology would be commercialized, and their declarations is usually off the target, but his research was realized in time, even faster than he had predicted. So I'm very lucky that I'm being given this opportunity to ask him questions. The first question: in standard cancer treatment, surgery, anti-cancer drug, and radiation were the three factors. What's the difference between PIT and the three traditional methodologies? The biggest difference is the targeting of cancer cells, and only the target cancer cells are removed from the human body. Because if it's a surgery, you end up taking away healthy cells as well. And anti-cancer agents, there is difference in terms of strength, but it also kills healthy cells. And also, radiation also does target cancer cells. At the beginning, I used radiation, but immunocytes are weaker than cancer cells, and immunity is considered to be the strongest protection against cancer, but that's killed along with the cancer cells. So without hurting those healthy cells, the cancer cells are targeted. This is the advantage.
For the patients, there's less of a burden. Yes, side effects are far less. So you take if you remove the normal organs, the function drops. And if we talk about immunity, immunity is one protective function. If you weaken that, a cancer will grow easily. So it has less of a burden on the patient's body.
You showed a slide of the photo as a result of the clinical trial. I was shocked. The cancer cell was beautifully removed and very cleanly. You were probably surprised. What was your first impression when you saw that result? We had been doing experiments. 1,000 animals, rats, were used for the experiments. So we were predicting the fast speed of the killing of the cancer cells. So it didn't come to us as a surprise. We knew the speed. But that and the therapy itself, it's just antibody and attaching radiation and then targeting cancer cells. But once again, we are anyway happy when it works on human body, but it wasn't a surprise.
So you got what you expected? Yes. We did experiments with animals, and the same thing occurred in human beings. Thank you very
でそして患者の皆さんにとって
The biggest concern for patients is what type of cancer, stomach cancer, breast cancer, pancreatic cancer. And in some other interview, you were saying 80% or 90% of cancer is curable through this therapy. Is that true? And what are uncurable types of cancer? Of course, in one drug, it's impossible to cover all types of cancer, so you have to have several types of antibodies. And as I had shown, first, the objective is to destruct the cancer cell. And there is huge diverse types of cancers, so you would have to have in place so many kinds of antibodies. Economically, that's not possible, but first, you can have a set of antibodies that would work for major cancers. So 5 or 6 antibodies would cover 50 to 60% of all cancers. And also, it's the protection of cancer. In other words, to get rid of the cells that are reducing your immunity level. So by having this two-phased antibody and combining those two antibodies, one that targets the cancer and one that targets the cells that would weaken your immunity, you would be able to cover many patients. And I guess 80% or 90% of the cancers would be covered with 10 antibodies.
You said 10. You mean 10 antibodies? Yes. Antibodies to cancer. There may be 6 to 7, and there may be 2 to 3 against immunity. If you have that, I think we can cover the cancer to the extent that I mentioned. Antibody is a word perhaps which is hard for the layman to understand. What is the role of the antibody? How do they discover cancer and bring the drug to the cancer cell? Well, cancer itself is such that it is generated where it shouldn't be, in places where it shouldn't exist. So cancer itself has a different kind of protein from the surrounding cells. And it generates protein which can become a target, which is different from the surrounding cells. We flash light on the spot, and the treatment is confined to that place. The cancer cell and the other surrounding cells, all we need to do is just distinguish between the two. Antibodies are such that if there's a target on the surface of the cancer cell, what attaches itself to it is the antibody. So to the extent that we flash light, in the area that we flash light, if there's an element that allows us to distinguish between cancer cells and other cells, then we can treat cancer.
In other words, it's like a courier company. You carry drugs to the homes of the people. Something like that, but the home called cancer could be, for example, how should I put it? The postal code. You would have to select the final digit number for the postal code. And then if you have that same final digit in the postal code, the same antibody A will be delivered and marked. So first, the cell you want to kill is marked, and then light is imposed, and then only the marked cell would die. That's how it works. So to a certain extent, you would aim to deliver it to a certain house, and everybody would get the light, but only the house that is marked will be killed.
So it's a two-tier treatment system, yes. The first application is without toxic agents. It's just marking. So once it's marked, well, you may have something that's marked. Without the NIR light flashed, it will not break unless you flash it with NIR light. So within the area that you have applied light, you can break the cancer cells. So we can control. And if we don't do anything, before you know it, it doesn't become toxic. And the antibody and the chemical all are ejected outside of the body, so it has no toxic effect on the body.
夢のようなピンポイント
It's a dreamlike pinpoint therapy, but are there cancers that would reject this therapy? The location where light would be radiated has to be the location where there is cancer. But in many cases, like leukemia or bone marrow cancer, it's difficult to identify the place of the cancer because you don't know where in the bone marrow the cancer cell exists. And the bone is made with substance where light it's difficult to project the light. And it's difficult to emit light inside the bone, and you don't really know where in the bone the cancer cell exists. So those are some of the types of cancer that is difficult.
No.
You have patients with that kind of cancer, so if they hear what you said, I think they would be disappointed. But blood cancer is such that recently anti-cancer drugs are quite effective, so that's a merit. And also, if it's that type of cancer, cancer of the blood, well, if we can't use this approach, I'm sorry about that, but there are other therapies. With respect to cancers that are difficult, for which there is very little therapy, you feel that this may be one solution to that. There's such a possibility?
Right. Solid type of pancreas cancer or lung cancer that occurs in organs, those are some of the difficult types of cancers, and where there is metastasis. And even without relocation, metastasis, maybe you weren't able to identify that metastasis at that stage, then the physicians would struggle. So this could be a breakthrough there.
Thank you very much. Earlier on, I also mentioned that the mouse experiments may work, but the same effect may not be obtained with regard to human beings. Very often we're disappointed, but is the PIT also applicable, effective on humans? As I have just shown, we mark the cell, and if you apply light, then before you know it, the cancer cell breaks. We don't use anything that's biological, that it breaks so quickly. It's like hammering the cyber, so it's a physical approach. So any kind of cell, regardless of the host, if it's an ordinary cell, if you mark it in a certain way, you can break it. With the mouse, you certainly have therapies that work well with mice, and with regard to humans, it doesn't work well. Of course, there are biological differences. If you use biological approaches, it may work on mice, and it may not work on human beings. But this is not biological. Cells are such that they are the same from bacteria to human beings. It's the same structure and mechanism. And so the cyber membrane, the membrane is absolutely essential for the cell to be a cell. It's surrounded by membranes. You scratch the surface of the membrane, so any kind of cell would die. So we treated mice, and indeed, initially there were views to the effect that, and people criticized that it may not work on human beings. But for our part, we felt that if it breaks cells, so it's the same with all species. And actually, between mice and human beings, it doesn't look like there's a major difference. So there's no difference, I don't think.
In other words, you target the cell membrane, so you can expect the same phenomenon. Targeting cell membrane versus the scratch on the membrane being physical. In other words, it's not using some kind of chemical and do something genetic to cause genomic change or mutation to create different type of protein. That's not the case. We aren't using the functionality of cell. It's just something physical, and antibody being attached. And by applying light and energy, we scratch the cell membrane. It's not biological, so the difference of mechanism between mouse and human is irrelevant.
I'm looking forward to what we can expect from this. Recent cancer therapy is very expensive. Recently, there's a drug which is very much talked about. Is this therapy also very expensive? And Japanese patients, how long will it take before Japanese patients can receive this kind of treatment? Could you talk about the specifics? I'm a scientist, so with regard to data, there may be, I can say something with a 1% margin of error. But if you make scientists calculate money, we can be off by 1 digit or 2 digits even. We're not very good at calculating money. And that's where the venture company representatives come into play. Maybe we should talk to them about it. So let me call upon one representative of the company. Mr. Mikitani, can you take off your Rakuten badge and change it to the A badge and come up here? Yes, Mr. Mikitani, could we have you up on the stage? No, I don't know why I'm being called. I'm taken by surprise. The patients are very keen to know when they'll be able to receive this kind of a treatment. And they're also worried that it may be very expensive since it's a very innovative treatment. To the extent that you know, could you tell us?
But before that, why am I up on the stage? Dr. Kobayashi was doing something really wonderful and amazing in the United States. And I got to know about his research. And I thought that this is a breakthrough technology that would change the future of mankind. I was a layman in medicine, but because I'm an amateur in medicine, I thought, oh, this is something physical. So just marking and breaking, oh, that's great. I was impressed, but my friends who were better versed with medicine would tell me that many research works on mouse, but not on human body. But I was told that it works on human body. And ever since, I've been supporting, in my individual capacity, the development under Dr. Kobayashi. And at the moment, FDA clinical trial phase 2 is underway in the US. And we hope to begin clinical trial in Japan as well. And preparations are underway so that we can begin shortly. And if possible, at the same timing, on global basis, we want to launch a product to the global market as expeditiously as possible. As far as the pricing is concerned, there are several factors and issues, and people criticize about the price in many ways. But the reason I've become involved in this project is because I hope that this would provide cure to as many cancers as possible at minimized cost. So, of course, appropriate level of cost is an important factor, and enabling as many patients as possible to enjoy this treatment is important.
Aspirion was shown on Dr. Kobayashi's slide. What kind of a company is it? I'm sure a lot of people do not know, so could you explain about this? In the case of the United States, it's different from the situation in Japan. NIH and the NCR, National Cancer Research Institute technology is licensed out to ventures, not large pharmaceutical companies, but it's licensed to venture capital companies. That's the general flow. The large pharmaceutical companies, if it's with just one project, it comes buried. But the ventures are very keen to commercialize, and so they become desperate to work on this. And that's insight on the part of Dr. Kobayashi. NH as a whole, with regard to this technology, decided not to give it to large pharmaceuticals, but to venture companies. And they've licensed out to venture companies. And I found these companies, and I've been providing funds, almost the sole existence, providing funds to these companies. With regard to this company, it's located in San Diego. And we've established a Japanese branch of this company. Rakuten involving itself in medicine, that's something that we find rather surprising. Could we hear about your resolve as an entrepreneur? We can take risks with regard to something that's not visible. With the 20 years ago, when nobody bought internet, we knew that the internet shopping would be mainstream. We built a lot of new businesses, and this time as well, I think it's a similar situation, a very similar situation. I think this is a major, large theme. It works theoretically, so I believe it will be of use. And whether it's Google or other IT companies, they are eyeing medical, a fusion of medical and ID. That's occurring very, very quickly. In Silicon Valley, that is the flow at the moment.
先生からじゃあ
Dr. Kobayashi, can you speak about the collaboration with Rakuten and what strength that would give you and your expectations towards Rakuten? Well, if you look at a traditional existing incumbent company who already has drug license, there would be conflict within the company. In other words, they already have something in their existing product line, so there could be conflict. But in the case of Rakuten or Aspirian, they've just picked this particular therapy and nothing else. So they're just running on that one singular avenue to cure the patients, to which I'm extremely thankful. And also, this is something that cannot be done without money. And we started from scratch, but because of the general support, Ms. Nagayama, as you mentioned at the very outset, in a very short period, when we expected 5 years to have to take until the clinical trial was completed, we were able to complete in 3.5 years. So I continue to solicit your continued support. Thank you. I know that there are so many other questions. I have so many other questions, but I hope that further progress will be achieved in the research so that this therapy is offered to patients, especially Japanese patients. Thank you. Dr. Kobayashi and Mr. Mikitani, thank you very much.